Hi, so this is the axon and the axon potential presented to you by Marissa, Kim, and Gigi. Here we will be explaining to you the parts of the neuron along with its functions, the difference between myelinated and an unmyelinated axon, the steps of the axon potential, and we will be giving you a graph of how that works. We hope you enjoy. Hi. Alright, so before we get into the nitty gritty, before we start explaining the functions and all these vital things you need to know, it's important that we know the structure and the functions of all these different parts of the neuron. So to begin, we're going to start with the dendrites. So the dendrites are those little yellow gangly looking dudes, the ones that look almost like roots or fingers. In simple terms, they just receive signals from other cells. So what they are projections from the cell body that receive information from other neurons called presynaptic neurons or from the environment. So another way that I kind of like to think of it is they almost resemble little fingers, and the way I think of it is, think of when you touch a desk. You touch it probably more with your fingers. Your fingers are what's feeling the desk. They feel the texture of the desk, whether it feels like it's going to give you a splinter, or you feel like it's soft, or the desk is grimy. You have these little messages coming in. That's more or less what these little gangly dendrites are going to do. They're going to receive signals from other cells. They're going to transmit these messages that need to come in that are essential for the neuron. Okay, so next up we have the neuron cell body. So if you look at where those little gangly dudes, the dendrites, are attached to, the rest of that yellow area, that's the neuron cell body. It organizes and keeps the cell functional, and it contains the Golgi apparatus, the nucleus, all the normal organelles. It's not very different from other cell bodies, the bodies of other cells that we've studied in the past, so it shouldn't be too hard to remember. Next up is the cell membrane. Once again, this is nothing new. This is very similar to a lot of other cells that we've probably studied or of from other biology classes. Just your typical cell membrane surrounds, protects, and supports the cell. Okay, so next up we have the axon hillock. So this part, I don't know if you can see the blue looking tail dude. So slightly above that, closer to the cell body, this is the axon hillock. So this is where it begins to become an axon. It controls the initiation of an electrical impulse based on the inputs from other neurons or the environment. It can sum up the total signals received, both inhibitory and excitatory signals. If this part's important, if this sum exceeds the limiting threshold, the action potential is triggered, which is kind of what this whole PowerPoint is about, which is very, very important, and we will discuss in a few later slides. Next is the nodes of Van Veer. This is very important. Personally, I found it a little bit confusing. So if you look at that tail-looking dude, it looks almost like the tail of a scorpion, where the blue is. So we're not looking at those blue spots, but we're looking at the spots in between it, those little gaps, okay? So this allows for the diffusion of ions. That's just putting it simply, okay? So pretty much there are gaps along the myelin sheath that covers the axon of the neuron cell. It recharges action potential, which runs along the axon. Because the myelin sheath is largely composed of an insulating fatty substance, the nodes of Van Veer allow the generation of fast electrical impulses along the axon. Without it, the action potential would propagate slowly, as the sodium-potassium channels would be disrupted. So this is really, really important. Without this, a lot of functions would not be completed within a timely manner, or the body wouldn't be able to function the way it needs to. You wouldn't be able to move your hands as quickly as you do. Okay, so if the nodes of Ranvier are the little yellow spots, those gaps that we just talked about, the Schwann cells are those blue areas. So pretty much they're glial cells that form and produce the myelin sheath on the axons outside the brain. So this is mainly for the peripheral nervous system. The opposite of it would be the oligodendrocytes, which are for the central nervous system. These are really important because this is what allows electrical impulses to transmit quickly and effectively along the nerve cells. Sometimes they can be damaged, which will slow impulses down. It should also be noted that Alzheimer's patients, the Alzheimer's pathology easily induces the myelin breakdown and the loss of the myelin sheath. So I'll go on to explain more the difference between a myelinated and unmyelinated axon and why it's important. But in short, without there being the myelin sheath, you cannot do a lot of the things you want to do. Babies can't walk if their axons never become myelinated. That's why Alzheimer's patients, their axons are myelinated, but as they begin to regress and the myelination is broken down and damaged, they slowly begin to lose that myelination. It'll make sense in the next few slides. The little purple thing in the center, this right here is the nucleus. It's very similar to any other cell that we've studied. Once again, it just controls the entire function. Very important, but pretty basic. The axon. So this is what we've been talking about when we've been referring to the myelin sheath and the Schwann cells, and this is where a lot of the studying will take place, zooming in on that axon and what's going on here. In short, this transfers signals to other cells and organs. They can be small or a couple of feet long. They carry electrical impulses that are the means of communication within the brain and the body. Alright, this is the one we've all been waiting for, the myelin sheath. In short, it increases the speed of the signal. And for the rest of the explanation, which is a bit lengthier, but it makes sense, I promise, it is the insulation for the neuron. It can help keep neurons from touching each other and messages interfering with one another. As we explained, they're constantly receiving messages, storing messages, sending messages, receiving messages. That would be pretty messed up if one message in your body got mixed up with another. It would just be a mess. They are formed by oligodendrocytes in the central nervous system, or Schwann cells, in this example here, in the peripheral nervous system. They are made up of protein, 20% of it, and lipids, the other 80%. It increases the rate of conduction of an actual action potential along the axon. Not all neurons are myelinated, and the speed of neuronal signal conduction is based on degree of myelination. More myelin equals faster action potential. Saltatory conduction is the action potential can jump from nodes of Ranvier to node of Ranvier. This is all really, really important, but once again, we'll talk more about the myelin sheath and the difference between myelinated and unmyelinated axons. And last but not least, we have the presynaptic terminals, or the axon terminals. They mean the same thing. So if you look at the very end, they look similar to the dendrites, but it's at the end, they're smaller. This is the presynaptic, or the axon terminals. So they're at the end of an axon, and they're the place where the electrical signal, the action potential, is converted into a chemical signal, the neurotransmitter release. In short, they form junctions with other cells. Alright, so before we get into that, next up we have membrane cells. So now I'm going to explain the difference between a myelinated versus an unmyelinated axon. So when an axon is myelinated, it's faster. And what I mean by that is it's going to kind of jump, the message that's being carried is going to kind of jump from each nodal space. And if you look on the slide right here, that little blue area between the two red hot dog looking things, or the beads if you want to call them that, that's called the nodal space, which is essentially a gap containing voltage-gated sodium. And it's going to make a jump from each gap to another, which makes the process quicker. The cloud of sodium carries the momentum of electricity, and this is pretty much what allows us to make the jumps between the gaps on the axon where it is myelinated. The fiber itself is also wrapped by Schwann cells, creating more myelin sheaths. All of this makes myelinated axons quicker. For example, when you walk, an axon will become more myelinated and tightly wound the more it is used. So think about when you're a baby. You don't really know how to walk. So as you begin practicing, these cells become more myelinated. And think about when you're walking now, you are able to walk with ease. You don't really have to think about it. You don't really stumble as much, considering maybe that you're not clumsy, but most people aren't going to stumble every 2 minutes like a baby does when they're walking, and this is because of the myelination that has happened on the axons. So here is what an unmyelinated axon looks like. As you can see, there's none of those bead-looking things. If there's a non-myelinated axon or unmyelinated axon, this would have to happen throughout the entire thing. The process always starts at the head of the neuron, then goes to the end. So because of those little bead-looking things, because those are not there, it's having to take the slow path to carry the message or to carry the momentum of the electricity. It's not able to make these quick jumps. It's almost like a path that's been built. There's no path that's been built, so it's taking longer for the whatever is trying to occur to finally happen. Going back to our baby example, when you're a baby and you're trying to learn to walk, or any baby's trying to learn to walk, they're often really uncoordinated and they stumble and they'll trip and they're holding on to things to try and walk. And that's because the path leading to the axon is unmyelinated. The baby has to concentrate and take only a few steps at a time before they fall. But the more that the baby practices walking, the axon will begin to become myelinated. So think of it as almost like a cinnamon roll. There's going to be lots and lots of tight rounds around the cinnamon roll. The more layers to the cinnamon roll, the better it is. The easier it is for you to eat it. Just like with the axon, the more myelination, the more sheaths, the more it's been tightly wound, the easier it is for you to do a certain task.
Hello everybody, this is Kim, and I will be explaining step 1, which is resting membrane potential. So basically I'm starting off with saying that cells have to be polarized before they become depolarized. The area around the membrane of resting cells is more negative inside than the outside. So inside you see here it's potassium, and as you can see outside Na+ sodium. And then cells have voltage, aka potential energy. These are all found in all living cells. This is especially important in neurons and muscle fibers. Inside of the membrane, it is more negative than outside of the membrane. So basically, this occurs because. So step 1. This occurs because sodium and potassium pumps keep sodium concentration higher outside of the cell and potassium concentrations higher inside of the cell. Number 2, there are a lot of potassium leak channels. There are also sodium leak channels too, but there are a lot of potassium leak channels. Number 3, gated channels are mostly closed/inactive. Number 4, unlike charges attract each other. So now moving on to inside of the cell. Number 1 would be high potassium concentrations, and number 2 would be negatively charged particles and ions. And then moving on to outside of the cell, high sodium concentration. So moving on to like an overall summary of this, basically it's just that the potassium door is open/leaky, as we can see in this photo. And number 2, the sodium outside neuron, potassium is inside neuron. Potassium is there because of larger anions. Number 3, the cell membrane is negative inside and positive outside. Number 4, the difference in ion concentration results in the resting membrane potential of the cell. And at resting state, the cell is polarized. So thank you. In the next slide, I'll be just describing the graph. Alrighty, so here I am explaining this graph. It's going to be quite short and simple, but later on I'll explain the peak going on. So basically, I'm explaining the number 1 here. Number 1 is resting membrane potential. As you can see, this graph, this is constant, is constant for a little bit at negative 70 membrane potential, which is basically resting membrane potential. The value remains constant until an external factor disrupts the cell membrane. Basically what it is, later on I'll explain why it peaks up. Thank you. Alrighty, hello. Now I'm here with this different image. I'm talking about depolarization. So basically, the gated sodium channels are open in response to ligand or voltage change caused by stimulus. Sodium diffuses down its concentration gradient into the cell. As more positively charged particles enter the cell, the inside of the cell becomes more positive, plus 30 membrane potential. Inside of the cell becomes more positive / less negative. So as you can see in this image, the sodium is going into the cell, and causing this to become a more positive environment for the cell. So an action potential begins when depolarization increases the membrane voltage so that it crosses a threshold value. At this threshold value, voltage-gated channels in the membrane open, allowing many sodium ions to rush into the cell. This results in less negative charge inside of the cell compared to the outside. Thank you. Alrighty, so now on step 2. Previously in the other slide, how I was talking about how sodium rushed into that cell, causing it to be a more positive environment. So previously, when it was just potassium, it was more negative, and sodium rushed in there into that gate, more positive. Boom. So basically, or less negative, however you want to say it. So previously in step 1, resting membrane potential, and how I explained how it was resting at negative 70, resting, but then you see how it spikes up, boom, where the number 2 is at. So basically, it's just less negative now, and this peak happened because of the sodium rushing into that cell, causing the environment to be less negative / more positive, and that's how it is shown in the graph. You can see this at the number 2. Thank you.
Okay, and this is Marissa. I'm going to be going over the last 2 steps. So here we're at step 3. The second we hit a membrane potential of plus 30, an automatic switch happens. The sodium door shuts because we can't let any more sodium in, and the potassium door opens, allowing potassium to leave, but eventually too much is going to leave. When the membrane potential hits the negative 90 spot, the sodium-potassium pump will turn on. In reference to our graph, we are here at number 3, which is repolarization. Everything previously mentioned happens, and the inside of the cell is now more negative. Step is step 4, where we are using active transport. We are moving from low concentration to high concentration using ATP. The pump activates at a positive 90 voltage to get rid of the sodium and stops at a negative 70 voltage. If there is any leftover potassium on the outside, it will want to come in through the door since it is positive. And lastly, here on our graph, it is number 4, hyperpolarization, where the potassium channels now slowly close, resting membrane potential is getting established, and the cell membrane is ready for the next stimulus.
Alright, and that would be the end of it for the axon and the axon potential. We hope you enjoyed our PowerPoint and that it will help you in further tests that you might take. Enjoy.